Bacteria reproduce through a process called binary fission, and this is how they multiply. And it is asexual, kind of like mitosis, not quite the same, but similar, where they're going to increase the number of cells by creating many clones. And this is a quick video. It shows how a single bacterial cell can become billions of cells in the matter of hours. But this process of binary fission does not create any genetic diversity. So the bacteria have to go through a different process in order to create genetic diversity amongst their individuals. Two of these processes are called transformation and transduction. Transformation happens when a bacteria cell takes up, so here's its main chromosome, it would take up a piece of genetic information from its environment and then take that into its cell and incorporate it into its body. And these are typically plasmids. They could be broken pieces of DNA from foreign other cells that were damaged in its environment. A lot of times during transformation, when it takes up a new plasmid, it's going to be something that confers resistance to an antibiotic or some kind of natural selective advantage. Another mechanism for increasing genetic diversity is called transduction. These are similar, so make sure that you can get these organized in your studying. Transduction is a type of horizontal transfer, meaning it goes from one bacteria to a different bacteria, and the vector between these is a virus. So here's the first cell, so this is the donor, and what happens is this gets infected by a bacteriophage, right, a virus that affects bacteria, and in the process of breaking up this cell's DNA and making new viruses, it makes a mistake. So instead of filling this virus with virus DNA, it filled it with the original bacterial DNA. So when this virus goes and infects a new cell, it's going to insert bacterial DNA, and then we might see some gene switching or crossing over occurring at the chromosome, and then we end up with a recipient cell with a different gene. So this would be a complete accident. This is not something that happens technically on purpose. And we know that the virus is typically going to carry the genetic information from an individual of the same species to another individual of the same species, because the virus-host relationship is very tight. So let's do a little summary. In order for a bacteria to multiply, means it's going to increase the numbers of cells, but these are going to be clones, so there's no change in diversity. And the only process that does this is binary fission. The other advantage to sex is increasing the genetic diversity of the next generation. So that's not going to happen during binary fission. So we need to increase genetic diversity in some way. This does not increase the number of cells. So the number of cells does not change. The first process we talked about was transformation. This is when it takes up DNA, so it was taking up plasmids or other broken pieces of DNA from its environment. And transduction, which is a virus moving DNA from one individual to another. The last type is called conjugation, and this is when one bacteria is going to directly donate DNA to another bacterial cell. The last mechanism for increasing genetic diversity is conjugation, and so this is what you might think of more as like mating. This is going to be a one-way transfer, so it's always going from a donor to a recipient. What would happen is we would have a donor cell here, and then we would have a recipient cell here. The donor cell has a piece of DNA inside of its body called the F factor. And this might be in a plasmid or it might be in its chromosome. And so at some point in here, it's got the F factor. So this donor cell is going to make a mating bridge across to the recipient cell and then transfer this F factor to that recipient. And along with that, it might transfer other genes along with the F factor, which is the mating factor. When the F factor is a part of a plasmid, conjugation might look like this. So this is the F factor is a plasmid. The F factor is what allows it to make this mating bridge. So here we have our donor cell. It's got the F factor. It's going to make a mating bridge. A piece of that breaks off, and it's going to transfer it across the mating bridge to the new cell. As it does this, you can see it's building copies of that, the complementary strand. And the F factor strand that comes into the new cell gets built a complementary strand as well. So it doesn't actually replicate this gene before passing it. Each cell gets one strand from the original cell, from the original DNA, and then each cell makes its own complementary DNA strand to make sure that it stays double-stranded. So now at the end, they didn't actually exchange any chromosomal DNA. This one started out brown, it stays brown. This one starts out green, it stays green. All that they exchanged was this F factor plasmid. The second scenario of the F factor can be built into the chromosome of the donor cell. So cells like this are called HFR, or high frequency of recombination. And recombine, so combine just means mixing. This is where we're going to combine genetics from one individual to another individual. So here we've got our donor cell again. This time the F factor is here. It's ingrained in the main chromosome rather than a separate plasmid over here. It's going to start at this breakpoint right here and then break off some portion of the gene that's going to be transferred to this other cell. So we can see that from here to here was transferred over, and then this donor cell has to go ahead and remake that complementary strand. So there's no change to the donor cell. The donor cell is going to stay the same. The recipient cell gets this fragment of DNA here, and these are going to be from the same species, or they have the same genetic trait here. So this trait for A, you could think of this like two alleles. A plus is one allele, A minus is the other allele. And so there's going to be some kind of crossing over or genetic exchange of these homologous genes. And the end result, we end up with a piece of the donor DNA incorporated into the chromosome of the recipient bacteria. This extra fragment, it's not an actual plasmid, it's just going to get broken down. So this cell does not actually get the F factor. So this cell is not going to become a mating cell. This cell is still the only one that can be a donor. But it has changed its genetics. Note that we did not change the number of cells here. We started out with 2, and we ended up with 2. The only thing that changed is this genetic trait in the recipient cell. So why is it important for these guys to have plasmids? A lot of the time, plasmids are called what we call R plasmids, or resistant plasmids. So these will carry genes that make that bacteria resistant to antibiotics. And these can be taken up through a transformation. These can be passed through transduction, conjugation. So here you can see down here are the genes that cause resistance to antibiotics, so kanamycin, ampicillin, those are common antibiotics used. And then this RTF fragment is the F factor that's going to allow for this plasmid to be transmitted to other cells. And this is going to be beneficial as far as natural selection for this bacteria, because now if it gets into an environment where it gets exposed to antibiotics, it's going to survive.